All right. So,
Go, Peter.
get started. Maybe hold your questions to the end, so you can make sure your teacher can cut it off. You guys need to get to a quiz. I know you're excited for those kind of things. But, all right. I'm Peter, and I am a PhD student in that Ellington lab at UT. Just remember, hold that thought, and we'll get to it. But in my research, I take living things apart and then I put them back together in new ways. So when I say that, you might think of something like this. But like Frankenstein, but actually you don't have to go and like dig up dead body parts and sew them together. As an example, think of your favorite Chinese restaurant, or imagine you have a Chinese restaurant if you don't. And you really like the atmosphere, you really like their lo mein. There's just one thing, you just wish that this Chinese restaurant would also make tacos. Then it would be perfect. The question is, how do you get the Chinese restaurant to make tacos too? Now, would you cut off half of a taco restaurant and attach it to the Chinese restaurant?
No.
Probably not. That would be kind of expensive. Do you just get a bunch of tacos from somewhere else and stick them in the Chinese restaurant? Maybe. But what I think I might want to do is I find the recipe book of that Chinese restaurant and I try to get a recipe for a taco. Convince the Chinese restaurant to include that recipe in their normal recipe book. And then start making it with all the other stuff they make. So that's kind of what I do. Okay. So living things are like a restaurant in that they contain a set of instructions. But a living thing would be like a restaurant where the recipe book doesn't just say how to make the food, but it also says how to make the whole restaurant and everything in the restaurant, the people, the toilets, the microwaves, and it also tells them how to do their job. So the name for that instruction book in a living thing is a word you may have heard before, but it's genome. So in my research, I want to edit that recipe book. So I call it genome editing. So before I tell you exactly how I do that, I'm going to talk to you a little bit more about what a genome is. So a normal recipe book, it's written in words, it has chapters like how to make the different things. But the genome in your body is actually not like that. Okay? It's not written in words, but it does have letters. It has 4 letters. Those letters are another word you've probably heard before. DNA. 4 letters.
Oh, that's great.
Well, DNA in the word has 3 letters, yes, but the chemical structure, as I'll show you, has 4 letters. So, I made up a little animation to show you how this works. It does indeed, because I have to click on it to make it work. So this is a chemical, a molecule, very small, that is the DNA letter that we call A. Okay? So you can then attach another different letter to it, in this case T, and then another one. You can just attach these letters in a string going on down, and then eventually you can say, hey, you can kind of read this. You got A, T, T, G, A, C. It kind of has some meaning to it. And this just goes on and on and on for a long time. And then when you take another similar strand and pair it, then you get that double helix structure of DNA, which you've probably seen before. So this is the language that the recipe book what life is written in, is this DNA molecule. Okay, so that's DNA. So if you were to read your genome, it would look basically like this. It's a whole bunch of letters. There's 4 of them in there, A, G, C, T. That's what your genome looks like if you read it. And this is also really, really long, your genome. If you divided this up into 300-page books and read one of those books every day, it would take you 22 years to read your entire genome.
Just for one human?
Yes.
So the DNA, the alphabet, is just those 4 letters?
Yes.
But then that means that she would have to read all day nonstop?
Yes. Well, read 300 pages. You can read 300 pages an hour. That's fine. But we use computers to read it for us. And you also have trillions of copies of this book in your body. In fact, you have 120 billion miles of DNA in your body, which is enough to go around the Earth 5 million times.
And you guys have seen DNA. Remember when we took it from the banana?
Yeah.
With that little white
The what?
thread-like structure?
Yeah.
The DNA when we took it out of a banana? So that.
This? Yeah.
100, how many miles is it?
It's 120 billion miles of DNA in your body.
That's how much a banana has to?
Banana is smaller than you are, but like a plant that has as many cells as you have would have about that much, maybe even more, because some plants have pretty big genomes. That's also enough to go back and forth between the Earth and the Sun 700 times. Which is pretty cool. But if you're like me, what you really want to know is, how do I get in there and mess with this stuff? Okay? So, that's what we'll talk about now. And the thing that I use is something called a lock site, which is a sequence of DNA. It's only 34 letters long, and in this picture, those arrows, those are the lock sites. And so if you put them on either side of a region of DNA, and then you have an enzyme, where an enzyme is a little tiny molecular machine that's made out of protein. This enzyme called Cre will come and put those lock sites together, line them up, and then switch things around. And in this case, what happens is that region gets deleted out. Now, if you change the direction of one of those lock sites a little bit, then instead of being cut out, it will just flip it around. And we call this an inversion. And you can also use lock sites to insert things in. So if you have your make a taco recipe, or in a genome you call it a gene, you can use these lock sites to put that new gene into the genome. Now the problem is, the genome doesn't usually have lock sites in it, certainly not in the places where you'd like them to be. So the question I had is, how do we put those lock sites where we want them to be? Now there are various ways of doing that. Between you and me, I thought they were kind of a pain in the butt. So we wanted to come up with a new way to do it. And to explain that new way, there's one other thing I want to talk to you about, another word you might have heard before, similar to DNA, but it's not, it's RNA. Yeah. All right, so to explain RNA, so I mentioned that you have many, many copies of the genome in your body. But you probably know that your body is also divided up into lots of tiny little compartments called cells. And each one of those cells only has 2 copies of the genome in it. So each of your cells is like a giant restaurant where there's thousands of chefs who all want to make recipes from only 2 recipe books. They can't all use them at the same time. They all want to use them at the same time. So what do you do?
PhD.
What this cell does is it makes lots of copies of the parts of the recipe book that it wants to use. And then it distributes the copies to the chefs. So these copies are RNA. RNA is like a temporary copy of a piece of DNA so that it can be used a lot. And this says how to make lo mein in your Chinese restaurant. So the cool thing is,
What is lo mein? Who knows?
Chinese food.
You know what it looks like?
It's a noodle.
I don't like this.
Okay.
I don't like this.
Oh. Nice
They
segue.
get all their recipes and then they just make tons of lo mein.
Whoa.
Okay? And the cool thing is that there are some types of RNA that can actually insert themselves into the genome and make themselves back into DNA. We use a special type of that called targetron. And this was invented by another lab at UT, and they're really nice because you can basically program them to insert anywhere in the genome that you want. Okay? So all we have to do is put the lock sites on the targetron, and then we can put the lock sites wherever in the genome that we want. So for instance, we could put lock sites in to delete parts of the genome and make a smaller recipe book. Or we can use it to put in new genes, like our make a taco gene into the genome. And then when the genome distributes those recipes to all the chefs, some of them will get to make a taco recipe. So they'll start making tacos in addition to the Chinese food they were making before. So that's basically how it works. Right now we do this in bacteria, which are really small organisms that if you look at them under a microscope might look like this. This is E. coli, or like this. This is staph, which causes a lot of infections. One of the first things that I did, just as a test, and just to show that I can do it, was to put a gene into the bacteria that makes them glow green. And so this was my Christmas card a couple of years ago, glowing green Christmas tree of bacteria. But so the question then is, well, what useful things could we do with this? And when I say bacteria, you probably think of getting sick, and it is true that a lot of bacteria cause diseases, and it's also true that a lot of the drugs, like the antibiotics that we use to fight those bacteria, aren't working as well as they used to. So one thing that we could do is use this method to delete out the bad parts of these bacteria, and then use the harmless bacteria as vaccines to help protect us against the dangerous bacteria. That'll be okay. And another thing is we often these days use bacteria to help us make complicated biological molecules. For instance, if you have diabetes, you have to take the insulin hormone. And it used to be that to get that, you had to kill a whole bunch of pigs and then extract their pancreas and then extract the insulin from the pancreas. But nowadays, there are bacteria that people have designed to make insulin. So we just get it from bacteria. It's a lot cheaper for us that way, and the pigs are much happier as well. So what I'm interested in doing is applying this to getting bacteria that will make fuels for us, so that we can get fuels without having to rely on fossil fuels, which pollute the air and will one day run out. So that's what I do. You guys have any questions?
Oh, I got 3.
All right.
Nice.
Okay. What
philosophy, but basically you graduate high school, then you go to college for your undergraduate degree for 4 years. And then if you really, really, really want to do research, you go back to what they call graduate school for however many years it takes. And at the end of that, the degree that they give you when you graduate, you are now called a PhD. And like most of the people who teach at universities are PhDs, and most of the scientists who do research are PhDs.
And you get to be called doctor.
Yes, you do.
Okay.
Doctor.
And how do you
I
come
will
up
be.
with the letters? You know the letters and the names?
Well, those are based on the names for those chemicals. Like the A is the actual name of that is adenine, and the C, the actual name of it is cytosine. So they just take the first letter of the names that those things are, and they use that as the name. And the names came from the person who first isolated that compound and maybe figured out what its structure was. They just decided, well, I'm going to call this one thymine because I like that name, or whatever reason they had. And everybody else just used it.
Which part is weird?
I don't know. It sounds kind of beautiful.
That's how science works.
I think I heard you
Any
say
questions there?
that people make bacteria. Like, what do you say?
Well, they use bacteria to make things, yes. And, well, kind of like I showed, there's ways to force them, get other genes into them to make stuff. And there also was a case recently where a group kind of did make bacteria. They just synthesized the genome using a machine, and then they stuck it into a cell and got it to start making all the recipes that the genome makes, and got a bacteria that way.
I have a question. Do you use the same letters for plants, animals, and people?
Yes, all living
The same
things
le-
have the
yeah, the
same
same letter.
code. Well, I grew up in Idaho.
I grew up in Dallas.
I'm not yeah,
Not done.
good. I live here now too myself.
No,
I've been here
I'm
for 4 years or so.
a grown up.
That's longer than me. I've also lived in Utah and Nevada and Japan.
I'm a single mom.
They're shorter on average than you.
They're asking me questions about chefs or something.
Where was Kitty?
Kenny is not a chef, so
This is Kenny.
Ah.
So she's very concerned about kitties.
O sea, ¿cómo puedes
No, the inside.
Enthusiasm. How excited are you about that topic? About this presentation?
any other questions?
What is Jan overall? Joanna,
No,
you're
I want to
having a
know
question?
if we can make a handshake.
Sure. What was your question?
She's curious about how can you use some genes from animals to humans. This is the question. Combine the genes.
Well, that is actually an important issue. On the one hand, all of life kind of uses the same coding system to get from DNA to the end product of that recipe, which is like a protein. But there are often differences in how it's turned on and off and things like that. And so if you want to move a gene from one organism to another, you often have to do a lot of work in order to modify it to get it to work in a new organism, which is one reason why all this isn't a lot more common, because it's not always that easy to move stuff around. It takes
So we can say that one A gene from a pig to one A gene from a human is not the same substance, I mean the same material?
They are often very similar, and different genes are what they call more or less conserved, which means they may stay more or less the same between different organisms. And sometimes you can, take the gene from a fly and replace it with a similar gene from a human, and it will work just fine. And sometimes it depends on the gene, but sometimes it works and sometimes it doesn't.
So can we say that I'm just relating with the elements for the periodic table? That one oxygen from water, it can be that one oxygen from something else.
Right.
Right? Something like that, comparing.
It's kind of similar to that, but sometimes your oxygen will work in the new water and sometimes it won't.
Okay. Thank you.
But like that glowing protein, that wasn't from bacteria, right?
Yeah, that's originally from jellyfish.
So it worked for like.
And that works. They've made that work in like mice, plants, and all kinds of things. So that one's one that works. Yeah. I got a haircut very recently. Before that, my hair was like this. But now I look very respectable. Just for you guys.
Looks like you said.
How rude.
Referral.
When you ask about what was in this question,
Oh, where do you get your question?
Another question?
She has another question.
Okay.
Come on, Joanna. Try it.
I have a question in if there's other person.
Did you say that again?
She's thinking about the work that you are doing. I mean, the research that you are doing is the same thing that researchers use to clone animals or
Like
people.
clone
Well,
sheep?
not people now, but
Well, in that case, they usually don't change the genome that they're putting it in. They just take all the DNA and put it into a
from
new
or
cell.
from somebody else.
Yeah.
They make that exactly copy, but they are not like a playing or changing
Right. Well, there's
from one organism
the
to another.
the environment that the embryo is in as it grows actually affects the way the genes get expressed later. So there will be differences due to that. But this could be combined with that cloning technology in order to, fix whatever problems might be intentional.
Has anything ever been cloned?
Has anything ever been cloned? Yes.
Bro,
Huh?
I've never seen it.
Yeah. The first thing they cloned was sheep, and they cloned a number of other things since then.
I saw some kittens. I thought they cloned some kittens.
They have.
But not really humans, right? So far.
Not that's been publicly announced.
Okay.
There's still a big problem with the humans with,
Yeah, for
that they can't go.
They might kill
For
Yeah.
the laws, it's for the laws that human rights or moral or something.
I don't technologically, understanding-wise, there's some huge leaps that need to be made before that's feasible. I don't think it's going to happen anytime soon.
imagine
I
some
got
really.
on TV. They really did it that way.
No.
The server said that.
It is illegal.
Why is it legal?
Because if someone was really rich and they wanted to clone themselves so that they could take over the world, right?
That's not possible because you have to die. Everybody does.
Yeah.
Okay, but if you clone yourself, then that person could take after you, or you could. I mean, if you wanted an army of like superhuman people, you could just like clone a bunch of Arnold Schwarzeneggers. Like people, it could go very wrong.
That's a big like a couple of years ago.
They're just not sure yet.
Right now, most of the clones are
Oh,
often
thank you very much.
very sick. And so you might get like 1 out of 50 that actually lives to grow up. So that's not very effective for
If
doing.
you clone the one with like, you got a common clone, would he die too?
No. So it's like identical twins are actually genetically clones of each other.
Then which one would die?
Neither twin has to die. It's just like you have a twin.
But you know how the animal dies? The other one?
Well, everything dies eventually.
Mm.
Clones tend to be less healthy.
It's like making a copy, right? So the copy isn't going to be as clear as the original.
Right.
So there might be some more mistakes that would show up.
So basically, what I understand is, according with the research, is try to help more about the diseases or being more healthy.
Right. Diseases and producing chemicals that we need. And there is work on applying these targetrons to get them to work well in humans. The lab that invented them is working on that right now, and in that case, you could use this to cure genetic diseases.
Only genetic.
Basically, yes.
Let me tell you this, why I'm asking you this, because unfortunately I have a sister who had an accident about 10 years ago, and she's now quadriplegic. So I'm reasoning, we are researching and reasoning about that. Probably that kind of research can help a little bit in the future
Right.
for
Perhaps.
those kind of people.
Well, potentially, this is far off in the future, but potentially you could modify someone's genes to cause them to regrow nerve cells that had died or something like that. And actually, one thing that the company is working on right now is there's actually a gene in your body that if you delete that gene, then you don't get sick with AIDS if you're infected with HIV. And so if you could go into people who have AIDS and delete that gene, then you could cure them of AIDS.
They won't die if they take that away from them?
No, they do fine. There's people who naturally don't have it and they don't get AIDS if they have HIV.
I thought that you get that from like.
Good. That's interesting. Very interesting.
Well, you can transmission of bodily fluids will transmit AIDS, but what it does when in your body is it has to get inside your white blood cells, and there's a certain receptor, a protein on the outside of the cell that the virus binds to get in. And if you don't have that protein, then the virus doesn't get into your cell, and then you don't get sick.
So you could just be born with it?
People have been born with AIDS, yes. Yes, if your mother has AIDS, there's a danger that you could be born with.
How many years did it take to change the genome and let people?
How many years to do what?
Change the genes?
Well, it's more of a,
Yeah, like people and
oh, how long before we can do that in humans? Well, there are some people doing it in humans right now using different methods like that. That AIDS treatment is, there's a company heading to clinical trials soon on that, and treatments for other diseases. As for getting these targetrons to work in humans, they're working hard on it, and it could be anywhere from a year from now to never. So we'll see how it goes for them.
There's a difference between like researching a dead body and a human body. But like us, like.
It is a big difference because,
sometimes it's
well, it depends on what you want to know. If you just want to know what the structure of things is, it's easier to do it on a dead body because they're not going to complain when you cut them open and all that. But some things you can't answer unless you use a living body, like things about like neurology, like the kind of things Josh does with, you know, which direction is it going to go, and you know what neurons do what, and how is that transmitted. You have to use living cells. You don't necessarily have to do it on a person. You can use cells that have been taken from a person and just look at the cells.
Is that scary? Like, listen like the movie?
I don't get it.
You've done that before?
I personally haven't done it, but a lot of other people in the lab work on cells like that. A lot of people who do cancer research, they do a lot of their work on what they call immortalized cell lines, where you just take cells from someone's cancer and then you can just grow them up in a petri dish. And like a lot of the famous strains, like the person who had that cancer died 50 years ago, but their cells are still alive and being used for research in cancer today.
They never die?
The cells are immortal, yeah. I mean, you can't kill them, but as long as they have food and oxygen, they'll stay alive forever.
All right, any more questions? I think we need to wrap it up at this point.
Yeah, I have one question. No, I don't
I'm liking y'all's questions. You guys are asking really good questions, though.
Yeah.
No more questions?
Yeah.
Okay, Sydney?
What's the difference between a dead body cells and live body?
Well, at first, there's not much of a difference, but what your cells do is they work very hard to keep what's inside the cell very different from what's outside the cell. And that takes energy. You have to eat food and then burn it in order to use the energy to keep what's inside different from outside. And once you die, all that stops. And then there's nothing to keep the inside from being different from the outside. And so gradually, you just become the same as the outside.
Interesting.
Okay.